Excellent. Okay, welcome everyone to the PGC Worldwide Lab for December. Thanks for joining us today. Just a few notes while I get started. This meeting will be recorded and posted on PGC website, YouTube, etc. And so just be aware of that. And we will be taking questions after the presentation, and people can do their questions in the Q&A, in the chat, or we're happy to have people actually just ask their questions. So if you want to ask a question, just raise your hand and I will try to make sure I figure out who's raising their hand, and then you can unmute yourself and ask your question yourself. I think we should be able to do that. So we really welcome people just, verbally asking their question. And so let's get started. We are really lucky today to have Renato Polimanti talking and representing the PGC-PTSD working group. And he is assistant professor of psychiatry in the Division of Human Genetics and Psychiatry at Yale. And he is going to talk about causal inference analysis of post-traumatic stress disorder challenges and opportunities. And I was really glad that Renato agreed to do this because he has really led the work, I think, globally in terms of MR, particularly in relation to PTSD, but he's really done a lot of the most excellent work in terms of MR broadly with psychiatric disorders. So I'm really excited to be able to learn again from him today. So take it away, Renato.
Okay, thank you so much for the introduction. And now you should see my presentation. It is in presentation mode. So again, thank you so much. I'm really happy to talk a little bit of the we did in doing causal inference analysis on post-traumatic stress disorders. There are some challenges, opportunities that applies to PTSD, but also applies to other psychiatric disorders. So this is my conflict of interest, and this is my scientific interest. I always like to show these slides at the beginning. So my group at Yale, we are interested in biological psychiatry, human genetics, evolution, and computational biology. This is my group. I'm really lucky to work with them. Yes. And our goal is to understand the links between human genome and human phenome from multiple perspectives. So before going deep in talking about PTSDs and causal inference analysis, I would like to give a brief introduction about some concepts that we need to understand when we decide to do causal inference analysis on PTSD. So probably you saw these slides many times in many different colors and shapes and tastes. And so we know that we need huge sample size to identify variants associated with psychiatric disorders. And this is due to the high polygenicity of psychiatric traits, but not all psychiatric traits are the same. For example, you can see that for schizophrenia, you need a smaller sample size to achieve these amazing results. Instead, for depression, you need much more. For PTSD, we were here for when this paper was published. Now probably we are kind of here, so a little bit better. And so there are differences across psychiatric disorders, but more in general across complex traits. There is this really nice paper published a couple of years ago in Nature Genetics that shows of effect size across complex traits, considering both continuous traits and disease. And here we can see how lucky we are in psychiatry because our traits are the one that have the highest polygenicity. So, for example, when you look at childbirth length, you can see that the curve is kind of flat. So you have a lot of variants with a relatively, we are always talking about small effect size, but very larger effect size. So, but then when we go to the other opposite of the spectrum, we have neuroticism, which is highly polygenic. So you have most of the variants with really tiny effect size. And when we go to disease status, here we can see that, for example, Crohn's disease, inflammatory bowel disease, and ulcerative colitis, they have relatively large effect sizes. And because with relatively small sample size, they were able immediately to get hundreds of genome-wide significant loci. On the other side of the spectrum, we have depression, and which is highly polygenic. So that's the reason why we need that massive sample size to do gene discovery with respect to this disorder. Here we don't have PTSD because at that time there wasn't a large study of PTSD, but I'll show later what's the issue with PTSD. So let's do focusing more about polygenicity of brain phenom. This is something that Frank Wend from my group did. So we analyze the polygenicity of different brain-related phenotypes, so anxiety-related traits, brain imaging, cognition, neurodevelopmental outcome, depressive and related traits, substance use and related traits. And you can see that there are different degrees of polygenicity. So for example, for brain imaging and neurodevelopmental disorders, we have a relatively lower polygenicity degree. Instead, for depression, anxiety, substance use, cognition, these are all highly polygenic. And we also investigated which are the mechanisms responsible for these differences. And we observed that background selection, so evolutionary pressure, shaped the polygenicity of brain-related phenotypes. We already know from previous studies that this is something already present for other complex traits. But another thing that we saw in this paper is that also diagnostic complexity influenced the degree of polygenicity. And we saw this association. So now we can talk about PTSD, post-traumatic stress. So these are the DSM-5 Criterium. And the first one is for people that are not familiar with post-traumatic stress disorder, you need to a serious injury or a life-threatening event or experience or witnesses this kind of event, and then you need to develop a series of symptoms and there are an entire spectrum. And with all these criterion, we have more than 600,000 possible diagnostic combination, which is a lot. I think, if I remember correctly, PTSD is the second most heterogeneous psychiatric disorder. And of these more than 600,000 possible diagnostic combination, we have more than 300,000 that are disjoint combination. So they are diagnostic combination occurring among a set of symptoms without overlap. So you can have two individuals with both a PTSD diagnosis without any common diagnostic, any common symptoms. So this is why we have this phenotypic heterogeneity that corresponds to a genetic, an increase of the polygenicity of the traits. So even smaller effect size, because we need to analyze something that is common among things that are not really correlated. So let's take a look to what we know from the genetics of PTSD. So this is a figure about twin-based heritability. Here we have several psychiatric disorders from bipolar, 85%, to generalized anxiety disorder, about 30%. And so we have a kind of this kind of range. With respect to PTSD, we have a strong variability even with twin-based heritability analysis. First of all, we have a strong and well-known sex differences where female PTSD heritability is much higher than the male heritability, but also we have differences with respect of which of the specific characteristic of the cohort due to, for example, traumatic events that are specifically targeting that cohort. And another point is that, as I say, trauma is the first criterion for PTSD diagnosis. And trauma is also an heritable trait, and in particular, assaultative events like sexual trauma, sexual assault, witnesses, murdering. So we have a 20% heritability of twin-based heritability. Instead, for non-assaultative events like disasters, we have a non-significant heritability. So we have multiple components here that work together, and there is a strong variability in how the genetics is associated with PTSD with respect to external factors. So what about GWAS of PTSD? So there was an early GWAS of PTSD, the first one from the Psychiatric Genomic Consortium in 2017, but was still a relatively small sample size, only 20,000 subjects. But in 2019, there were 2 GWAS, one from the Million Veteran Program and one from the Psychiatric Genomic Consortium. And these were the first large-scale GWAS of PTSD. And we were able to identify several genome-wide significant loci and start to investigate the polygenic architecture of PTSD. But fortunately, we are moving forward, and now there is a new PTSD GWAS that was accepted in Nature Genetics and is available on preprint. So we were able to identify multiple genome-wide significant variants, considering different PTSD definition from a case control definition to different symptoms. And we were also able to identify variants associated in diverse populations, especially in African American participants of the MVP study. And so this is a summary, this figure is a summary about what's the status of gene discovery of PTSD. And we have the number of genome-wide significant loci with the case control, and we have like a relatively small number of genome-wide significant loci with continuous trait. We have more power, so we are able to identify more variants, but the total is about 15 genome-wide significant variants. And again, we have a strong genetic correlation across these different definitions. So these are basically the same variants that we observe. So let's take a look to the polygenicity degree of PTSD. As I mentioned before, among the most polygenic traits, there is depression and neuroticism, and the PTSD and PTSD symptom cluster. So quantity traits of PTSD, they are even more polygenic than depression and neuroticism. This is probably due to the phenotypic and diagnostic heterogeneity of PTSD. And so this means that we need, here we have the number of genome-wide significant loci with respect to the sample size that we expect in the future. And we can see how massive the sample size needs to be to investigate PTSD. So there is a lot of work to do, and it's going to be really challenging to identify a large number of genome-wide significant loci. Fortunately, we can investigate with the current datasets the polygenic architecture of PTSD. So this is a figure from the PGC-PTSD article published in Nature Genetics last year, and we compare the genetic correlation pattern of PTSD with other psychiatric disorders across different domains, so psychiatry, anthropometric, smoking behavior, reproductive outcome, aging, education, and cardiometabolic traits. So we can see these differences and different associations that PTSD has with other complex traits, and a similar analysis was done also in the latest MVP study, and we have the strongest correlation and also a comparison between PTSD and PCL symptom counts, so a quantitative traits of PTSD, and the strongest correlation, again, depression, tiredness, mood swings. And here we have, again, a comparison with respect to different phenotypic domains, anthropometric traits, behavior, circulatory traits. And we can see that there is a consistency between the binary definition and the quantitative definitions. And usually we see that the quantitative phenotype is more informative. Again, we are seeing all these genetic correlation between PTSD and other complex traits. And what this means that we have pleiotropy. This is not something new. We well know that human genome, there is a widespread pleiotropy, so variants associated with multiple traits, but we can distinguish two main types of mechanism, a shared pathway or a causal mechanism. So I know on Twitter people don't like to talk about vertical pleiotropy and horizontal pleiotropy, but I'm not that sophisticated, and I actually like this distinguish between these two mechanisms. So with the vertical pleiotropy, we have a genetic variant. This is associated with the first phenotype, and then the first phenotype has a causal effect on the second phenotype. So we have a causal effect here. This is the vertical pleiotropy, also known as mediated pleiotropy. Instead, with an horizontal pleiotropy, we have the genetic variant that is associated independently with the two different phenotypes. Obviously, these are the really simple examples about very complex mechanism. So genome-wide, we have kind of a mixture of these two components. So there are many different mechanisms, many different scenarios how these two things can be mixed up. So we can use Mendelian randomization to investigate vertical pleiotropy, and so have information about causal effect among complex traits. So a Mendelian randomization is similar to a randomized controlled trial. The principle is that variants and alleles are randomly distributed in the population, which is not completely true, because there are mechanisms like assortative mating that kind of are against this assumption, but let's assume that for now. Like similarly to a randomized control trial where people are randomized into different groups, so a group that is exposed to a medical intervention, a control group, then you don't adjust for all possible confounders, and then you check your outcome. Instead, here you have that the variants are randomly assigned the population, so you have a group allele, and then you have a control group. These are not associated with any confounder, and then you can investigate the outcome. So the Mendelian randomization is based on three main assumptions. The first assumption is that your genetic variant needs to be associated with your exposure of interest. The second assumption is that the genetic variant shouldn't be associated with any possible confounders that links the exposure to the outcome. And then the genetic variants need not to be associated with the outcome independently from the exposure. So basically, this means that the only way your genetic instrument is associated with the outcome is through the exposure. So this is the take-home message. It looks simple, which is not, because there are many possible ways why these assumptions are violated, and to apply causal inference to PTSD, we will need to violate some of these assumptions, at least partially. So recently there was an explosion of Mendelian randomization study, and this was due to the fact that we now can run Mendelian randomization using summary statistics. So summary statistics are available everywhere, and they are free. And the two-sample Mendelian randomization approach is relatively not computationally intensive. It's relatively quick. So we can have one exposure GWAS extracting the variants that we are interested in. Then we have an outcome GWAS, extract the information about the variants, and then harmonize these variants. So I'm being careful about the defect allele in both the datasets is concordant, and then if you are missing information, taking a look for LD proxies, and then you run your analysis, and then you do several sensitivity analyses to verify that there aren't possible outliers in your genetic instruments, that there is not heterogeneity, pleiotropy, and all the other kind of stuff. So it looks simple, but it's not, because there are many ways, and there are especially many methods that are available to investigate, to apply to sample Mendelian randomization. And some of these methods works better with respect to specific scenarios. And so the thing that you want to do is to apply multiple methods to be sure that your results is not method-driven, so that is not due to a specific weakness in the approach that you're using, but is consistent. So when we apply to sample Mendelian randomization to PTSD, we have several challenges. The first one is about the first assumption. The first assumption, as I said, is that the genetic instrument is associated with the exposure. As I said, PTSD is highly polygenic. So we have a limited number of genome-wide significant loci. These have very small effect size. So when we want to generate an instrument for PTSD, and we want to use only genome-wide significant variants, we don't have power, because the best that we can have right now is 15 variants, and it's not really powerful. So the first thing that we need to do if you want to work to do a causal inference analysis testing PTSD as an exposure, we need to relax our criteria to define the genetic instrument. So we need to use quote unquote a weak genetic instrument. And then we need to be careful about the issues that a weak genetic instrument can have. Other issues that we have when we apply Mendelian randomization to PTSD is that, as I mentioned, there are many things that can link a traumatic event to PTSD. And so there are many ways that you can have genetic variant associated with possible confounders, or also can be associated with the outcome independently from the exposure. So there are multiple things that we need to be careful about when we apply Mendelian randomization to PTSD. So this is kind of a framework that in my group we are using, and other people probably are using something similar. And the first thing when we are interested to analyze and test an hypothesis is to verify if we have good genetic information. So a SNP mate, the SNP heritability, and then test the genetic correlation. So and test the genetic correlation give us some information about how informative are these genetic datasets to investigate the association that we know is present for two particular phenotypes. Sometimes, and I have an example for PTSD that I'm not going to mention because I don't want to start a discussion about it. We know from the literature that there is an association about two specific phenotypes, and then you find the two powerful enough data sets that give you a good estimates of the heritability, and then you test the genetic correlation, and there isn't a genetic correlation. So for some reason, your observational study gives you a different outcome when you investigate genetic correlation. There may be several explanations why is that, and but the take-home message is if there isn't a genetic correlation, probably you don't have the power to do a Mendelian randomization analysis. So that's the first step that we do. The second step is that about the weak genetic instruments. Like when you have a small number of genome-wide significant variants that you cannot define like a genetic instrument including only genome-wide significant variants, we do a thresholding approach. So similar to a polygenetic score with the clumping and thresholding method, we use different threshold and going lower than the genome-wide significance, so and testing which one has the strongest power. And then when we find something that is working, we do our Mendelian randomization and we test our hypothesis using multiple methods and comparing the estimates, and then applying as many as sensitivity analysis possible to verify that there isn't heterogeneity or horizontal pleiotropy or other possible confounders. And if we observe, for example, heterogeneity, we try to verify if there are variants that are contributing to this heterogeneity. We try to remove the variant from the genetic instruments and rerun the analysis, and then we check if the result changed or not. If the result changed, so from a significant effect, a non-significant effect, this means that the outliers were contributing to the result. If the result doesn't change, it means that the outliers were not contributing to the result. So, and when we have a significant effect, the other aspect is, as I mentioned, pleiotropy is widespread. So when you test an exposure with respect to the outcome and you find a clean effect for your Mendelian randomization, you should consider the possibility that this effect is somehow mediated by a third variable. And so conducting a multivariable Mendelian randomization help you to understand if the effect that you're observing is really independent from other possible confounders. So I'm going to give you two examples that we did with the data from the PGC-PTSD workgroup. The first one is about PTSD and educational attainment, and the second is about PTSD and C-reactive protein. So there is a well-known relationship between educational attainment and PTSD, also with cognitive performance. There are several studies also in large cohort the observed association. There are also longitudinal studies that observed a specific direction. Unfortunately, there are longitudinal studies that they show opposite effect. So in some cases, PTSD affect educational attainment, and in other, educational attainment was affecting PTSD. So we don't know if cognitive performance and educational attainment are a pre-trauma risk factor or a trauma/PTSD outcome. So we can use genetic data to test this hypothesis. And so here we use PGC data. We consider two different release of PGC data, one including only PGC course, the second including only PGC plus UK Biobank, and then several traits related to educational attainment, cognitive performance, and also some possible mediators of this association. So using PGC data, social science genetic assessment consortium, UK Biobank, 23andMe, we started to run this analysis. So the first thing, as I mentioned, let's test the genetic correlation. And we have the strongest correlation for educational attainment with respect to the PTSD2, which has the largest sample size. We are using also PTSD 1.5 because with some methods like PRS and Mendelian randomization, we need to avoid sample overlap. And so we have a reduction of the power. And then for cognitive performance, we have a lower statistical power with respect to educational attainment for PTSD2. And we don't have significant genetic correlation for childhood intelligence and cognitive performance when we use the PTSD 1.5 dataset. And then we can do an analysis to estimate the ability of this genetic instrument to predict our outcome. So in this way, in the blue dots, we have PTSD is the outcome. These are the exposure. And we use, since these are powerful GWAS, most of them, except for childhood intelligence, we use genome-wide significant variance. And we can see that we have significant association between these genetic instruments and PTSD. When we do the opposite and use PTSD using a suggestive threshold, because we don't have genome-wide significant variance for the PTSD 1.5, we see that we don't really have the power to do this analysis. So, and here we can also see if we do kind of like a thresholding approach, including more variance, so relaxing our inclusion criterion, we can see that our power increase. So the strongest association was between highest math class completed and PTSD. And this was because this was 23andMe, this was PGC plus UK Biobank. So this was the best combination. And here we have our variant with effect on math class and PTSD and multiple methods. So we observe that with multiple Mendelian randomization approaches, we see a concordant effect. The thing that you need to remember is that these methods have different power and account differently for possible biases within the genetic instrument. When you do an analysis, you don't expect all of them to be significant. The one that usually is significant is the inverse variance weighted approach because it's the most powerful. The thing that you expect is that if there is a causal effect, is that this should be consistent in the effect direction. And then you need to run your heterogeneity analysis, MR intercept, MR pressure, and then MR RAPS over dispersion. So there are several sensitivity analyses to check if the effect that you see here within this variance is consistent and you don't have outliers. So in this analysis, we saw some outliers. So we removed the outliers from the genetic instruments and rerun the analysis, and we didn't see a difference. So in this case, we did several tests comparing two different methods with similar power, so the EVW and the MR RAPS, but also comparing the results across different data sets that included different samples. So we have the two PTSD data sets that I mentioned before, and then we have the educational attainment, including only social science and UK Biobank, cohorts, and then we have a larger educational attainment with social science, UK Biobank, and 23andMe, and then we have math class that includes only 23andMe. So with math class, and in this case we are able to use PTSD2, we see that there is no difference between the two methods, and also the second estimates, which looks the same, but this is without the possible outliers. We don't see any difference in the effect estimates, so the outliers were not contributing to the association that we were observing. And then when we remove the UK Biobank from our PTSD dataset, we saw a reduction of power, which is fine because we lose a large number of subjects, both in case and control. So the reduction of power was expected, but we saw a consistent effect with what we expected before. And then when we changed our genetic instrument using different dataset, we saw a consistency across the estimates. So this means that our result was not driven by a particular method or a particular dataset. Okay. What about the opposite? So when we try to do our analysis from PTSD to educational attainment, we try to include more variants, but we were not able to achieve a significant prediction. So we try to use a different approach, which is called MRAPS, which is a different method that is specifically designed to account, to conduct Mendelian randomization based on weak genetic instrument. And when I mean weak genetic instruments, I mean genetic instruments including variants where the association is not so defined like variants that reach genome-wide significance. So we did this test, testing both directions, because one evidence of causality is that you should have only one direction that is significant and not the other. So we tested both directions, including all the variants available in these GWAS datasets, doing, of course, clumping to reduce the correlation of the variants within the genetic instruments. So this is the genome-wide Mendelian randomization, so including all variants, all LD independent variants in the genome, educational attainment on PTSD, and we observe a similar effect estimate that we observed before using only genome-wide significant variants. And we don't observe pleiotropy within these genetic instruments, and then there are multiple corrections that you can make on this approach, and the results is pretty stable. When we do the opposite, we don't have a significant association between PTSD genetic instrument and educational attainment, but we have a huge pleiotropy within the genetic instruments, means that there are other mechanisms within the genetic instrument. And here we see that the results, when we apply more stringent correction, become even less significant. So right now we have an association, a potential causal effect between educational attainment and PTSD. And the thing is that we may have something in the middle that mediates the effect of educational attainment on PTSD. So we choose three additional exposure and conducted a multivariable Mendelian randomization. So if a Mendelian randomization is similar to a randomized control trial, a multivariable Mendelian randomization is similar to a factorial randomized trial, where you test multiple exposure and analyze their independent effect. So in our case, we choose risk-taking behavior, childhood physical abuse, and income. The reason why we choose childhood physical abuse is because actually we tested different kind of traumatic events and then choose the one that were the strongest and that then was not affected, his association with PTSD was not affected by possible confounders. So this is our multivariable Mendelian randomization, and we have our two methods, so the AVW and multivariable EVW. And so we can compare the estimates. So the dark blue is the effect not accounting for the other exposure, and the light blue is the effect accounting for the other exposure. So when we account the effect of trauma, income, educational attainment, we don't see a big change in the effect of risk-taking behavior. There is a little bit of power reduction, but this is normal because we are accounting for additional information. And so we see the same thing for childhood physical abuse, no difference. Income, no difference. For educational attainment, we see a big change. So from a negative effect, we see basically a null effect. And so this means that one or more than one of these other exposure is mediating the effect of educational attainment on PTSD. So here we tested then models with only one possible mediator. So risk-taking behavior, again, no change, but also no change in educational attainment. So this mediator was not risk-taking behavior. Childhood physical abuse, same thing, no change here, no change here. Income, there is no change in the effect of income, but then educational attainment completely changes the effect. So this means that our mediator is income. So, and if you think that income may be a better brain GWAS than educational attainment, we did some analysis to test the enrichment of brain mechanism in these GWAS, and we can see that educational attainment is a much better, is much more informative for brain mechanism than income. So there are other possible mechanisms that link income to PTSD, and so this is not related to educational attainment. So we provide really relatively strong evidence that educational attainment doesn't not have a causal effect on PTSD. So the second study was about C-reactive protein and PTSD. C-reactive protein is an inflammatory biomarker that is routinely used to investigate, for example, the risk of heart disease. This is a scale that is used in clinical practice to identify people with high risk of heart disease based on the CRP levels, but CRP is not specific. So increased level for CRP can reflect other possible phenomenon. For example, when we have a really high CRP levels, we may have other conditions like cancer, autoimmune disorders, or infections. But in addition to that, we know that inflammation is a key aspect of PTSD, and there are several studies that observe association between PTSD and C-reactive protein. So we decided to test this hypothesis, again, testing using the PGC-PTSD datasets and using the charge inflammation working group GWAS for C-reactive protein, and then possible mediators using UK Biobank data. And the first thing that we did is our analysis for the genetic correlation of C-reactive protein with other traits. And so here we have, again, socioeconomic status, income, protective, so negative genetic correlation and positive genetic correlation for the deprivation index. So again, here we have social support in blue, which have a negative correlation with a C-reactive program, and instead traits related to trauma response or trauma exposure, they have a positive correlation with the C-reactive protein. Here we don't have enough information to conduct an analysis using only genome-wide significant variants. So in both directions, we use weak genetic instruments doing this thresholding analysis, and so defining both for CRP and PTSD genetic instrument only with variants that have suggestive effects. And this is the results. We did the analysis not only for C-reactive protein and PTSD, but also with the other traits that we were investigating. And so at the end, we were able to identify effects that were consistent across multiple methods, and they were not confounded, at least with the outcomes that we got from the sensitivity analysis. And so these were the association that we observed. So with respect to PTSD as an outcome, we saw possible causal effect for C-reactive protein and the prevention index, and a protective effect with the loved child, so childhood support and household income. However, we also saw opposite effect direction. So also CRP had an effect on on household income, the genetic instrument, same thing with felt loved as a child, and also with respect to CRP, PTSD, and household income had an effect on this trait. So we have a bidirectional relationship, which means that probably there isn't a causal effect, but rather there are shared mechanisms. However, we also tested that social independence among these different effects. So we tested felt loved as a child, so social support, and deprivation index, and CRP, so three traits reflecting three different domains. And what we observed is that socioeconomic status was partially mediating the effect of social support and CRP on PTSD, and the same thing was also present in household income. So these two traits were mediating the effect of social support and CRP on PTSD. So this means that, again, socioeconomic status has an important effect on PTSD. So more recently, we decided to go big because we saw, and there are several studies that show that educational attainment and socioeconomic status are associated across the brain phenome with several psychiatric disorders and behavioral traits. So we decided to go big and test all of them. And so this is a figure about the genetic correlation between psychopathology, psychosocial factors, educational phenotypes, and socioeconomic status. And using, starting from this information, we conducted a comprehensive analysis testing how this trait, so educational attainment and socioeconomic status, is affecting our ability to investigate the polygenic architecture of brain-related traits. So if you are interested to know more about this paper, there is the preprint on medRxiv, but if you wait probably 1 month or so, you should be able to read the final study on Nature Human Behavior. Hopefully it will come out soon. So take-home messages about using Mendelian randomization for PTSD. We can use this approach, although we have to slightly change the approach, because these are highly polygenic traits where we have few genome-wide significant variants. So sometimes we have to use weak genetic instruments. So the important thing is comparing the results across multiple methods. If multiple datasets are available, testing differences across multiple datasets, and also applying appropriate sensitivity analysis to confirm that our results are not biased by possible confounders. Another important aspect is the application of multivariable analysis, because most of the times when I see Mendelian randomization studies, people are testing one exposure and one outcome and making conclusion about it. However, we have a widespread pleiotropy, so we need to test the possible effect of other potential mediators and test if the effect that we are observing is actually not mediating by another trait that may have a completely different meaning in understanding the relationship. So I want to thank again the people in my group. I'm lucky to work with an amazing group of people, and again I'm also very lucky to work with the PGC and MVP investigator on studies related to PTSD and related comorbidities. And mainly I would like to thank Joel, Karestan, Murray, Caroline, and Dan, which are the people that I work the most, and of course our funding agency. Thank you so much for your attention.
Great. Thank you, Renato. That was terrific, as always. We had one question posted in the chat, and I had to restart my computer. So there was something else I missed. They'll have to repost. So Abe Palmer, you can see it. Abe Palmer posted, there might be sexual differences for highest math class completed for sociological reasons. Are you able to account for this given the large sex difference in PTSD?
So that's a really good question. The problem is that right now we have limited power to split our datasets. Like PGC-PTSD has an equal representation of both sexes, and so it would be really interesting to split and test possible differences. However, if we split the dataset, which it's currently enough powerful when we combine everything together. But if we split it, it's not going to be powerful enough to do this kind of analysis. Of course, in the future, it would be really interesting to do this kind of analysis. For example, we are just finishing to analyze the relationship between PTSD and anthropometric traits. In this case, since the sex differences with both traits were very large, we were able to find sex differences. So yes, it's definitely a good point, and hopefully we will have more data soon.
Yes, that's great. We always want more data. We need more data. Other questions, folks? We don't see hands up, but people can jump in with questions. Just looking. Okay, Howard, using PRS with a large fraction of the genome essentially guarantees pleiotropy. How does one deal with that?
Yeah, that's another good point. Like again, also when you are using only genome-wise variants, and the fact that our GWAS fortunately are growing and growing, and the number of variants that reach genome-wise significance are growing, you have the same issue there as well. Again, the important thing is that you check your results. So when you use a weak genetic instruments, you have to test your hypothesis, see if you see a causal effect, and then check within the instrument which are the variants that are contributing for heterogeneity or for possible confounders, and then remove those variants. And then retest your analysis and make sure that you don't have the heterogeneity anymore. So you are using a weak genetic instrument, but you are removing the variants that have the strongest association with possible other phenotypes. And so they may be more responsible for the horizontal pleiotropy.
Good. Yeah, that's a really interesting question. Other questions, folks? Let's see, talking. I'll ask a question. What do you, I was thinking of the MVP, the slide you showed on the genetic correlations, and I was curious, where do you think some of the most interesting places you're going to go next with PTSD and trauma and MR?
So another thing is that,
yeah.
Another thing is Mendelian randomization is not the only method to do causal inference analysis. There are other methods, and the nice thing it would be to combine all these methods and try to see a convergence. Right now we are exploring the consistency across. Like right now, for the previous study, we tested the multiple MR methods, and we saw some kind of consistency. Instead, when we use a completely different approach, we don't see the same consistency. So we are trying to understand better what are the different mechanisms and why we are not observing the same causal effects. Again, because causality and pleiotropy, they are really complex to investigate. So your method needs to model many possible different scenarios that link two different phenotypes, and within your genetic instruments, you have many variants that may be associated with these two different phenotypes for different through different pathways. So we need to do a more careful job in, first of all, standardize the field, because right now there are so many different methods and different ways to apply the results. So kind of the future that I see, which is not only related to PTSD, but again, PTSD is so challenging because it's so polygenic, and we have so few genome-wide significant variants. So it's kind of like a test to see if we can be able to leverage more the polygenicity of the trait, but at the same time make the results more consistent and more reliable. So I saw also another nice
question. Yeah, I mean, so something that's just following up on what you just talked about is which other causal methods, can you mention a couple examples?
So, for example, another method that we are using is the latent causal variable analysis, which leverage the genetic correlation and try to understand if there is a latent variable that is mediating this genetic correlation. And what we are observing is that in some cases we have a consistency with Mendelian randomization, and in other cases we don't, and we want to understand why. Because the result should, if there is causality, it shouldn't be due to the method, but should be an objective result. So why we are not observing these two different things. Like in the Mendelian randomization, we do all our sensitivity analysis and we see a clean causal effect, but then we move to the latent causal variables and we don't see the same thing. So we need to understand better why these two approaches are modeling different certain combinations of analysis.
I agree. You said very clear. So Monica asked, do you anticipate that your results may differ in the future as GWAS become available from more diverse ancestors?
Yes, that's really a great point, especially because all of our results are based only on European ancestry datasets. So definitely it would be interesting to understand how these translate to other ancestry group where we have both genetic variability, so a different genetic architecture, maybe also a different polygenic architecture, and especially for the African ancestry, where the genetic structure is so different from non-African population. We have so much more diversity, so there may be difference also in now the polygenicity degree that we have across complex traits. This is something that it's my hypothesis. I would really like to talk with other people about that. Not now because we don't have time. So and another point is also that we have environmental difference across ancestry group. Like in the US, we are well aware of socioeconomic status differences across different ethnic groups. And so when we test these with genetic instruments, what we are going to observe, this is absolutely an interesting thing that I'm really looking for. Hopefully we will get data soon to explore differences in applying Mendelian randomization and other causal inference analysis in other human populations.
Thank you. I apologize. People hear my dog barking, but she's. So what are recommended approaches on identifying biological influences of traits such as SES and educational attainment on PTSD biology?
So I think like is for sure one thing that sometimes is missing from Mendelian randomization analysis is that we can also go inside the genetic instrument and try to understand the biology and, like, for example, do gene enrichment analysis to see, for example, if there is a specific pathway that is mediating that association between two possible phenotypes. So right now, most of the Mendelian randomization analysis, they are focusing on testing possible causal effects, but then we could also combine like functional enrichment analysis to understand also possible molecular mechanism, because again, probably we don't have any clear, clean scenarios where we have only vertical pleiotropy and horizontal pleiotropy. Is a mixture of the two things. Of course, there will be some associations that are more clean, and there are other associations that are not that clean, like in the causal, the difference between vertical and horizontal pleiotropy. So definitely, like adding more analysis to understand the molecular mechanism could give us a better understanding of how these two different mechanisms interact with each other.
Excellent. Yeah, and that's and then sort of related to that, are you going to study whether these effects mediated by socioeconomic status are related to social stratification? I think you kind of alluded to that some, but
yes, again, like I'm really interested in what socioeconomic status means from a genetic point of view, because I don't have a I did several, in my group we did several tests, and with Karestan and other people we did several analyses to understand what is socioeconomic status genetics, because it is not the same thing than socioeconomic status that is observed, and also like there are possible confounders that we may have, like related to population stratification. In our analysis, we didn't see population stratification as affecting our results. Another aspect that I didn't mention, but we analyze, is the assortative mating that we have, for example, for educational attainment, socioeconomic status. And this is kind of a violation of the Mendelian randomization assumption. So the variants are randomly distributed in the population. Instead, when you have a assortative mating, the variants are not randomly distributed in the population. So we accounted for that. So in our case, for the relationship between socioeconomic status and PTSD, we didn't see that as a possible bias, but still remain the questions: what is socioeconomic status from a genetic point of view?
That's interesting. I wonder, Renato, this is a bit of a random question from me, but has anyone looked at assortative mating related to trauma history or PTSD? I mean, there's a lot of sort of non-genetic, other kind of clinical work which might posit that.
We didn't run the analysis, but I would expect that we should see a sort of mating there, especially like also for the other components of possible factors related to PTSD, like risk-taking behaviors, neuroticism. So it would be really interesting to how assortative mating shape all these possible contributors to PTSD and how they affect the genetic correlation. And yes, definitely. That's
interesting. I hadn't thought of I just wasn't really thinking until you mentioned that. Other questions? That one. I don't see any hands up. Any last questions, folks? I think so. Okay, well, that was great. Thank you so much, Renato.
Thank you.
And, thanks everyone for attending and for the great questions. And we look forward to seeing where this goes. Like Renato said, we'll have more data, in the next 3 to 6 months. We always want more data. So if anyone has any PTSD data, come join us. But, thank you. Take care, everyone. Be safe and be well. Bye-bye.
Bye-bye.
Excellent, Renato. Thank you so much. Really great. Lots of thanks in the chat. Thanks, everyone. Take care. Okay. Bye-bye.